Last time I talked about some basics of structure of DNA, which I think probably were mostly review for many of you. But today I want to talk about the proteins involved in DNA. I got a little bit of a start with that last time, talking about DNA polymerase I. And so I want to come back to that right now and say some more about that. As I showed you last time, DNA polymerase I has a structure that looks kind of like a hand. And this hand is really designed to hold that double-stranded DNA that's there. It turns out it doesn't hold it real well. That's one thing about this hand. The hand doesn't close. The hand is an open hand, and we can imagine that the DNA lays or lies inside of that hand well. But if the DNA pops off, the hand doesn't clinch. The hand doesn't really hold it. So it's just a little place for the DNA to lie. Right? We'll see that's actually a factor for DNA polymerase I. All right. DNA polymerase I has several different activities that I talked about, and I want to spend a little bit of time talking about those activities. So let's discuss them right here. All right. So DNA polymerase I has three different enzymatic activities associated with it. Three different enzymatic activities. The first activity is pretty straightforward. It's a DNA polymerase, meaning it makes DNA. And when it makes DNA, it catalyzes the formation of phosphodiester bonds. So it makes phosphodiester bonds between adjacent nucleotides, and it has the ability to read the opposite strand to know which nucleotide to put in. That's the number one activity. So it's a 5' to 3' DNA polymerase, making phosphodiester bonds. The second activity of DNA polymerase is one I started to refer to last time. It's called the proofreading activity. Well, you know what a proofreader does. A proofreader is somebody who reads something that you've written, something that you've typed, and looks for any errors that might be associated with what you've done. And if they find those errors, they correct them. And the proofreading activity in DNA polymerase I does exactly that same thing. It goes along and it looks to see, okay, well, I thought I was supposed to put in a G. Does that thing that I put in there fit? In which case, I probably got the right thing. Or does the thing that I put in there not fit? In which case, oops, I made a mistake. Okay? So that's the question that it asks. If it fits, then it says, okay, I go on to the next nucleotide. And if it doesn't fit, it stops. And it has to remove what it put in, which means it has to break the phosphodiester bond that it created to hold that nucleotide in place. So what it does is it backs up. It literally goes backwards, and it chews out that nucleotide as well as 3 or 4 nucleotides ahead of it, just to make sure it got everything. When you go to erase something after you've been proofreading, you don't always erase just the letter that you got. You maybe erase the whole word. So this thing erases the whole word. Several of the bases that are in there. This is a result of a second activity of the enzyme that's referred to as the 3' to 5' exonuclease. That's proofreading. Now it's 3' to 5' because it's starting at that 3' most nucleotide and working backwards in the 5' direction. Completely backwards to the way that the polymerase itself was working. Okay? So it goes backwards, chews it out, and then it says, okay, let's try it again. And then the polymerase kicks back in, and it goes forwards. Does that make sense? Okay. So the proofreading activity is a critical function. Because of proofreading, DNA polymerase improves its fidelity, that is the accuracy with which it works, by about 100-fold. Meaning it's about 100 times more accurate, maybe 1,000 times more accurate, because it has proofreading. How do we know this? Well, we know of DNA polymerases in things like HIV. HIV has a DNA polymerase. We'll talk about it later. That's an odd DNA polymerase. And it's odd in several respects. One of the respects in which it is odd is it does not have proofreading. What that means is that the DNA polymerase associated with HIV makes a lot of errors. And those errors that it makes turn out to be, to the virus's benefit. You've heard how HIV eventually becomes immune to drugs used to treat it. How does it do that? By virtue of the fact that its polymerase is making errors for it, and some of those errors turn out to be beneficial, and those beneficial errors are selected for. Okay? So there's two enzymatic activities associated with DNA polymerase I. The third activity was rather surprising. Okay? You might say, okay, everything needs to proofread, right? You might say, we obviously have to have a DNA polymerase. But the third activity, when you look at it on the surface, you kind of scratch your head and go, what the heck? Okay? The third activity is another exonuclease. However, instead of working in the 3' to 5' direction like a proofreader, it works in the 5' to 3' direction, the same direction that DNA polymerase activity is working. Now, why in the hell would a DNA polymerase have an activity that destroys the very thing it likes to make? All right? Well, I'm going to save that answer to that question for just a minute. All right? I'm going to save that answer to that question for just a minute. The answer turns out to be rooted in the way that DNA is made. Now, I should point out to you that or remind you that I'm talking about DNA polymerase I. This is a polymerase that's in E. coli. It has activities that are very similar to polymerases in other organisms. Okay? But when we talk about DNA polymerase I, we're talking about the one that's in E. coli. Okay? All right. Well, I said I was going to talk about DNA replication proteins. The second DNA polymerase, or the second DNA replication protein I want to talk about is called primase. When Arthur Kornberg discovered DNA polymerase I, as I said, he thought he had the right one. Turned out he didn't, at least for the main replication. DNA polymerase I does make DNA inside of cells, but not very much. We'll see why that happens. Okay? Not very much. It turns out that when you make DNA, if you take DNA polymerase that you've purified, and you take a strand that you want to copy, and you put in 4 DNTPs, and you say, go forth and make DNA, nothing will happen. If that's what you put in the tube, nothing will happen. The polymerase will sit there and twiddle its thumbs. No DNA will be made. Why will nothing be made? Because a DNA polymerase always requires a pre-existing strand it can work from. That pre-existing strand is called a primer. All right? Well, how do you get a primer there if you have to have a primer to start making a primer? Because I just told you DNA polymerases need to have something there, so how would you get the first one there? Turns out the first one that's there isn't DNA. The first one that's there is RNA. And how does it get there? It gets there by action of an enzyme called primase. Primase. Primase is an RNA polymerase, meaning it makes RNA. And primase does not require a primer. So now if I take that strand of DNA, and I take primase, and I take the 4 DNTPs, and I take 4 ribonucleotides, all right? Then I will see a primer be made, and I will see DNA be made. Okay? If I don't put the primer in there, nothing's going to happen. If I don't put the primase in there, nothing's going to happen. All right. If I don't put the ribonucleotides in there, nothing's going to happen. But if I do all these things, I can make DNA. All right? Now notice the orientation of these strands. I didn't say this the other day, but I want to make sure, and I'm sure you guys have heard this before, that nucleic acids in a duplex are always in an orientation we call anti-parallel. Anti-parallel. meaning that 5' to 3' of one strand is opposite to 5' to 3' of the other strand. Yes? Why does DNA polymerase need a primer? Because the activity is only able to extend. There's no way to put down a first nucleotide. There's nothing in it that allows it to do that. It will not work. Yes?
So you're still copying from the same strand that the primase started?
It's correct. So it's copying the red strand. It's extending the black one. Right. Right. So they're copying the same strand. All right. So that's what primase does. Primase, as I said, it's a very specialized kind of RNA polymerase. We'll talk about other RNA polymerases later. But this is a very specialized kind of RNA polymerase. Well, now we begin to get the idea about why it is that DNA polymerase I has this 5' to 3' exonuclease. Let's imagine that I'm replicating a DNA in the cell, and I start out with an RNA primer. Do I want my finished product to have that RNA primer? Absolutely not. Because there's going to be a problem down the line. If I go to replicate this strand and I go backwards this way, when the DNA polymerase hits there, it's going to go, that's not DNA. I can't copy that. I can't go backward. I can't go and make that. So the RNA has got to get removed and it's got to get replaced by DNA. What we will see is that bacterial cells have circular chromosomes. They're a circle. So you see it linear here, but imagine that this circle goes all the way around, and that DNA polymerase starts here and it comes all the way around, and it gets back over here, and it gets back to about where it started, and it hits this RNA. You know what it does? That's where the 5' to 3' exonuclease starts chewing away what it made. So the 5' to 3' exonuclease is essential for removing RNA primers. In fact, that 5' to 3' exonuclease is the only enzyme in the cell that is capable of doing that. There's no other enzyme in the E. coli cell that can do that. Okay? So all three activities of DNA polymerase I make good sense. Okay? Everybody got me? Okay, so primase makes sense. The DNA polymerase I makes sense. I mentioned the fact that Arthur Kornberg found the wrong enzyme. How did he know he found the wrong enzyme? Well, he discovered that when you put the DNA polymerase with the DNA, that it went onto the DNA, but it didn't stay on the DNA for very long. It would go on, it would make 10 or 20 or 50 or maybe even 100 nucleotides, and then it would fall off. And if I wanted to replicate the entire strand, the entire circle of DNA that an E. coli cell had, a polymerase had to fall off, come back on, fall off, come back on, maybe have many different polymerases that were involved. And people knew from looking at structures of replicating DNA in an electron microscope that it looked like a polymerase went on and it stayed on. It didn't come off. So DNA polymerase I didn't meet the criterion. That was one hint that it didn't work. It wasn't the right one. The second was that when you looked at the rate that the DNA polymerase I was working, it wasn't working nearly fast enough to replicate DNA at the rate replication was occurring in the cell. Okay? What's that? Yeah.
You talked about the one that Calvin falls over. It was DNA polymerase I.
DNA polymerase I. So DNA polymerase I is an example of an enzyme we call processive. On, off, on, off, on, off. That's processive. It proceeds from one to another, to another, to another. Well, there's a reason why it does that. We'll see in a bit. So after they've spent all this time isolating this enzyme, they bop themselves in the head and go, there must be another one. So they went and they went back. And these guys, when they isolate these things, they will take E. coli. How many people here have worked with E. coli? It's the active ingredient in shit. Okay? And that's where the smell comes from. Okay? And so this is really not fun to work. So you have this, large pile of cells that are sitting here, and they're rather gross. And now your boss has just told you, well, guys, we got the wrong one. Let's go back and dig in. right? What's your attitude, right? You think you have a bad job, okay, or a bad attitude. Go back and let's go looking again. So they go back and they find another DNA polymerase. Voila, we've got it, right? Nope. Wrong one again. They called it DNA polymerase II. DNA polymerase II turned out to be involved in repair mechanisms, damaged DNA and so forth. It's a relatively minor DNA polymerase. DNA polymerase I, the reason they found it first was it's the most abundant one in cells. Cells have a pretty fair amount of DNA polymerase I. Well, now they're scratching their heads, right? They go back and they say, well, there's got to be another one. And so they had to take this large mound of cells, and they had to make it much larger to get the very trace quantities of the polymerase that was actually there. That polymerase they found after a lot of work, and it became known as DNA polymerase III. Now, I'm going to say some things about DNA polymerase III, but I just wanted to let you know that DNA polymerase III is the enzyme that is responsible for most of the replication of the E. coli chromosome. And yes, we call it a chromosome, even though it's not really technically the same as our chromosomes. Okay? And it had the right properties. It went on and it stayed on. And the reason it wasn't so abundant as DNA polymerase I was because it went on and stayed on. If something goes on and stays on, you don't need too many. You need one going this way, you need one going this way, and basically you're there. It turns out that in an average cell, there's about 6 or 7 copies of this enzyme. Not very many. As opposed to thousands of DNA polymerase I. So it wasn't surprising they didn't find it when they first looked. This thing also worked at the right rate to replicate DNA. This polymerase worked at the rate of putting bases into making duplex DNA of 1,000 nucleotides a second. Now, you've heard of enzymes that I've talked about that work at 1,000,000 per second. So 1,000 per second doesn't sound so hot until you realize what that polymerase is doing. It's attached to a DNA. And we'll see later how it gets attached to the DNA. It is sliding along the DNA. It is reading the other strand. It is grabbing the right nucleotide. It is forming a phosphodiester bond. It is checking to see did it put the right one in, and then it's moving on to the next nucleotide. And it's doing that 1,000 times every second. That is remarkable. Yes? Yes, so his question is, does DNA polymerase III have a proofreading activity? It does. But it does not have a 5' to 3' exonuclease. So it has a 3' to 5', but not a 5' to 3' exonuclease. How fast does Pol I work? Pol I probably works at about, I would say 1/10 to 1/100th of that rate. 10 to 100 nucleotides a second. Still pretty good, but nothing like polymerase III is doing. Okay. Well, how in the world does polymerase III manage to do that? Polymerase III turns out to be, first of all, a much more complicated enzyme than polymerase I. It has many subunits. Polymerase I only has, it's a dimer, basically. Okay? Meaning two identical subunits. Polymerase III has many subunits, and they have some specialized functions. So let's talk about, first of all, the other proteins that are involved in DNA. Then I'm going to talk about some of those multiple subunits of polymerase III. Okay? Actually, maybe I won't do that. Maybe let's talk about the subunits of polymerase III first. That's the way we'll stay on the right subject. Here is what polymerase III looks like. Okay? You can see it's got 2 identical sets of subunits, meaning it's a dimer. Okay? We are not going to talk about the individual functions of all of the subunits. In fact, I'm only going to highlight one right here. Let's look at this, though. Okay? Here is the main part of the enzyme where catalysis is occurring. It's called the core. You see there are 2 cores. Look at that structure that's there. That's just like that structure we saw in DNA polymerase I, where that hand in the palm and the DNA had a place in which it could sit in that palm. But I told you DNA polymerase I came off. meaning that the hand didn't clamp down on the DNA and hold it. It turns out DNA polymerase III has a clamp. The clamp is not part of the hand. The clamp is this guy right over here. This thing labeled beta 2. It's known as a beta clamp. And you can see it's a ring. And this ring starts out like this, open, put DNA in it, and it closes. And so when this clamp closes around the DNA, the polymerase is held to the DNA. That's how it gets on and stays on. Everybody with me? So the beta clamp is a very, very important subunit of polymerase III because it tells us how polymerase III remains on the DNA. We refer to polymerase III as progressive. It gets on and it progresses all along the DNA. It doesn't pop on and pop off. It progresses. Okay. Now, I'm going to explain why this is a dimer in a bit. It's actually a little complicated, okay? But I'm going to explain why it's a dimer in a minute. This clamp loader right here is responsible for taking that clamp that's open and closing it around the DNA. That's what the clamp loader does. Okay? Okay, questions about this? Yeah? Okay, so question is, I'm talking about E. coli. What does human or other cells, what do humans or other cells look like? We actually have a more complicated picture in our cells. We actually have 7 or 8 different polymerases. E. coli turns out to have 5. There were 2 more that were discovered later that were odd polymerases. We don't talk about them here. I'm not going to talk about them. They're really not significant for our purposes. But when we look at our setup, even though we have a more complicated setup, we have exactly the same activities within our proteins. We have enzymes that facilitate polymerase proofreading. The polymerase will proofread. There are subunits that help that happen. There are subunits that will remove primers. Okay? Now, in our cells, we have different kinds of DNA. For example, we have regular nuclear chromosomal DNA. We also have mitochondrial DNA. And so they have their own polymerases, and it is a more complicated picture. Okay? But suffice it to say that the activities that we see in E. coli, we see in every cell on the face of the earth. So there's some very big commonalities in the way DNA replication occurs. Yes? So his question is, how does the DNA polymerase I remove the RNA and not remove the DNA? Okay? The answer is it removes a little bit of the DNA. Okay? Let me go back, and it's actually a good question. Let me just fill you in a little bit here. So if we look back at the priming that's happening, remember I said that it comes all the way around and gets back over here, and now it says, oh, look, I can start chewing this up, and it starts chewing up this RNA as it gets along. All right? And it gets over here, and it doesn't stop right there. It probably chews in here a ways. But this, folks, is why it's very important that it falls off. If it didn't fall off, what's going to happen? It's going to chew the whole bloody thing. Right? So this processive nature of DNA polymerase I actually works to the cell's benefit. It doesn't get on and stay on, and so you lose a little bit. It loses a little bit of DNA, but it doesn't lose much. What would happen if it fell off before it degraded the primer? Okay? What would happen is another one would come in. Because they come in, they go off. Remember I said it's on and off and on and off? So if it came off before it degraded the primer, another one would come in and do its thing. Yep. Yes?
I'm curious. So you said that we have mostly in our E. coli has mostly polymerase I.
Yes?
Even though the polymerase III seems to do better job, only it doesn't have 5 to 3 exonuclease. Then why do we have so many polymerase I versus polymerase III that is much faster?
Okay. His question is, why are there so many more copies of DNA polymerase I compared to DNA polymerase III? I'm going to answer that question in just a minute. Okay? You're anticipating it. It's also a good question. Before I do that, though, I want to tell you a little bit more about the proteins, and then I'm going to show you what's called a replication fork and answer that question, okay? Okay, so you've seen some of the proteins that are involved in the replication of DNA. I want to talk about a really cool one right here called helicase, okay? Let's look at what this does first. Oh, what I do, don't do that to me. All right.
Surprisingly, the ring is not six-fold symmetric, but is slightly split, create nucleic acid duplex into their component strands, using energy from ATP hydrolysis.
Okay, so what this is showing you, let me stop here. What this is showing you is that when DNA replication occurs, somebody's got to pull the strands apart. Somebody's got to pull the strands apart. The strands don't magically come apart, and DNA polymerase I or polymerase III, or whichever polymerase we're talking about, is busy enough that it doesn't have to pull the strands apart. It actually has something called helicase that allows it to pull the strands apart. So here's what it's showing you, the helicase in action.
Separate nucleic acid duplexes
See it?
into their component strands, using energy from ATP hydrolysis.
Okay, so it's using ATP to pull those strands apart. Now, here's something really cool, all right? The helicase has got to pull the strands apart, and it's got to pull the strands apart fast enough that the polymerase doesn't get slowed down. Uh-oh. The polymerase wants to go like a bat out of hell. The polymerase wants to go 1,000 nucleotides a second. That means that the helicase needs to be unwinding strands at the rate of 1,000 nucleotides a second. It does. So it's unwinding strands at the rate of 1,000 nucleotides a second. 1,000 nucleotides corresponds to 100 turns of DNA, because there's about 10 base pairs per turn. So it's doing it's unwinding 100 turns of DNA per second. If you multiply that 60 seconds in a minute, you discover that this thing is going at 6,000 RPM. That's faster than your car's going, folks. This is a little tiny molecular motor that's unwinding DNA at the rate of 6,000 RPM. Why doesn't it break? Yes. Why doesn't it break? It doesn't. But it stays ahead, and that's why when you saw the figure that I showed you here of the primase in action, I'm sorry, of the, was it here? No, not here. When I showed you the polymerase III in action, it said helicase up here. You can see that it's up ahead of where the activity is. It's unwinding in advance of everything else. Yes, question? It's a good question. It's actually his question is the helicase spinning around the DNA or vice versa or some combination of that? It turns out that the way that it unwinds is a sort of a stepping mechanism that is more complicated than I can go into here because it's a biophysical problem. But if you want to think about it as literally pulling apart the strands, that's the best image I can give you. Yes?
I have a question. So I know that the backbone of the DNA is kind of protecting the DNA, but at the same time, when we pull the strands apart, it still happens in aqueous solution. Why those hydrogen bonds don't right away happen with...
Oh boy, this is a great question. So his question is, DNA, if you look at a double helix, the phosphates and the sugars are on the outside, very hydrophilic, and the bases are very hydrophobic and they're on the inside, but when I pull these guys apart, now I've got hydrophobic bases that are interacting with water. What the hey, Ahern? You've already said this isn't good, right? Turns out that there's one of the proteins that's involved in replication of DNA called single-strand binding protein. All right? It's there for a couple of purposes, one of which has just been described. It covers up bases. The second thing is it protects that strand. Your question earlier was, why doesn't it break? If the strand breaks, the cell is hosed. So the cell goes to extraordinary measures to keep that very tender, that very sensitive strand from breaking, and it has this single-stranded binding protein that grabs and gloms onto it very well. It turns out you might think it'd be in the way of the polymerase, but in fact the single-stranded binding protein actually helps the polymerase. Okay?
Yes, but now it's not a confusion. Then if you know that Helicase works so much faster to allow the DNA polymerase not to be stopped, then how fast those single-strand binding proteins should all be removed so DNA polymerase doesn't stop? It's so many things that would stop it.
Yeah, so what he's coming to grips with, his question is 1,000 nucleotides a second, you've got proteins coming on, you've got to have proteins coming off. This basically requires an amazing amount of orchestration. It's a mind-boggling amount of orchestration, all right? And it works. It's the best answer I have for you. Yeah, I mean, what you're seeing, I mean, his sort of amazement here, right? He's shaking his head, and that's my reaction to it too. We're going to see there's even more complexity of the replication of DNA that makes this orchestration even more remarkable. Okay? There's still a couple wrinkles yet, but this does all work well. Okay, now, so we've talked about helicase, all right? We need to talk about a topoisomerase. What in the heck's a topoisomerase? Let's imagine I am that helicase pulling apart the strands. If I start pulling apart the strands at this point, what's going to happen up ahead of where I'm pulling apart the strands? Is it going to be more twisted or less twisted? More twisted. I'm going to create more twists. And I'm already worried about breaking DNA. I don't want to break my DNA. And the more twists I put into it, the more likely something like that's going to happen. Cells have an enzyme that helps to relieve that tension. It's a necessary enzyme. It's called a topoisomerase. And what a topoisomerase does is it changes what we call the superhelical density of a DNA duplex. It changes the density of it. What's the density? Well, I said that Watson and Crick's DNA was about 10.5 base pairs per turn. That's density. If I increase it to 15 base pairs per turn, I have really wound it down tight. And if I decrease its density to 5 base pairs per turn, I have loosened it. And it turns out that both of those are issues for DNA. Topoisomerases can increase and they can also decrease the topological density, the superhelical density of a DNA. Now that turns out to be important. They can do both. Okay? They don't usually bring it to 10.5. They usually bring it to something else. That creates something we call superhelices, superhelical structures. I'm going to show you that in a bit. All right? But just keep in your mind that topoisomerases do not usually bring it to 10.5. 10.5 base pairs per turn is what we call relaxed DNA. Most DNA in cells is not relaxed. Okay? But it's also not as tight as it would be if we didn't have a topoisomerase. Because if we didn't have a topoisomerase, that DNA would be in a knot. And it would knot at the rate of 6,000 RPM. Okay, you get the idea. All right, so we've got a topoisomerase. By the way, the topoisomerase, there are several topoisomerases, but the topoisomerase that is ahead of the place where replication is occurring is called DNA gyrase, G-Y-R-A-S-E. That is a topoisomerase. So DNA gyrase is a topoisomerase that's ahead of the place where replication is occurring. Yes? How is it working? Let me come back when I talk about topological considerations and answer that question for you. Okay, so her question was, how does it do it? And I'm going to save that for a future talk. We're not done with all of the proteins yet. Okay? Sliding clamp. That's the beta clamp I told you about that was on the polymerase that's helping to hold it onto the DNA. It can open and it can close. Once it closes, it's going to hold that DNA polymerase III to the DNA. Beta clamps will not work with polymerase I. Beta clamps will not work with polymerase I. They will not hold polymerase I to the DNA, and that's why polymerase I comes off. They will only work with polymerase III. Okay, yes?
So why does DNA polymerase III have two beta clamps?
Why does polymerase III have two beta clamps? Okay, we're getting close to me answering that question for you. Okay? Very good question. All right, notice I haven't talked about DNA ligase. I'm coming back to that for a reason. Let's look at what happens during the replication of DNA. This is what we call a replication fork. The place where replication is occurring occurs at a fork because we can see the two strands being peeled apart. This figure is not very accurate because it makes it look like there's two different DNA polymerases, but in fact, they are both subunits of that dimer. All right? It turns out that both strands are being replicated at the same time. So that polymerase is replicating this strand, and the polymerase is replicating this strand. Now, this turns out to pose some logistical issues for the polymerase. The strand on the left is pretty easy to replicate. You start at an end. You start at a 3' end, and you keep going forwards, 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 and you keep moving upwards, and the helicase keeps unwinding for you, and you make this guy in one giant piece. But I want you to remember something I told you on Wednesday. What I told you on Wednesday was polymerases always, always, always only work 5' to 3'. This guy is 5' to 3' just fine going this direction. What's the orientation of this strand? It's opposite, right? That means replication can't be going upwards. It means replication's got to be going downwards. And though this makes it look like this is two nice enzymes where one of them is sliding down here and the other one is sliding up there, that ain't the way she works. This guy replicating this strand can only start replicating as a new region gets opened up. This one over here opens the region. So the replication of the leading strand causes these strands to unwind, and it opens up a new region that can now be replicated and go back down this way. Okay? So what happens is the leading strand, which is the one on the left, is made in one piece. The lagging strand, which is the one on the right, has to start over and over and over and over. It's made in tiny pieces called Okazaki fragments. And every time it makes one of those little tiny pieces, it needs to start with an RNA primer. Every one of them starts with an RNA primer because, oh my goodness, here's a new piece of DNA. I can't start with DNA. I've got to make an RNA primer first. So it makes an RNA primer, and then DNA polymerase III fills in a ways, and we have a series of RNA DNA, RNA DNA, RNA DNA, RNA DNA, RNA DNA going along there. And it turns out that the polymerase can't join them together. Those little pieces are individual fragments. They have to be joined together by something, and they're joined together by that enzyme I didn't show you, or didn't talk about, which is the DNA ligase. It links the pieces together. But wait, it can't link the pieces together because you can't put an RNA with a DNA. You don't want to, right? What has to happen? Polymerase I has to come in, and polymerase I has to chew out that RNA. And in answer to your earlier question, why does the cell need so many polymerase Is but not so many polymerase IIIs, is because it's got a lot of Okazaki fragments, each one of which requires a polymerase I. Because every time you've got an RNA, you've got to remove it. Yes? Very good question also. What determines the length of an Okazaki fragment? I'm going to show you a video, probably not today, but I'll show you a video on Monday that will show you the complexity of this, and part of it is how fast the polymerase works. Yes?
Yeah, so it seems like, okay, so we say, for example, this Pol III, and it's 4,000 nucleotides per second, including this process that
Yep.
seems like one arm works very fast, another one very slow. So without this lagging strand, probably could go in much faster.
The leading strand goes much faster than the lagging strand. You are correct. Yep. And this causes some very interesting issues. It causes a structure to form that we call a trombone. I'm going to show you that on Monday. Okay? I see a lot of hands. Whoa, okay. Yeah, here.
Does DNA ligase only work DNA to DNA?
Yeah. So our question is, does DNA ligase only work DNA to DNA? So DNA ligase only works DNA to DNA. So that RNA primer's got to get removed, and then the pieces are joined. Other question here.
With all the additional binding and enzyme activity for the lagging strand, how much more energetically expensive is it to synthesize the lagging strand than the leading strand?
Oh boy. Okay, so good question. How much more energetically, or first of all, is it more energetically expensive to synthesize the lagging strand than the leading strand? The answer is it is. Because you're making a lot of RNA that's getting removed, and every time you do one of those, you're throwing things away. So the lagging strand is much more, I shouldn't say much more, but it is more energetically expensive than is the leading strand. The answer to the question is what percentage of that is made as RNA, and I don't have an answer for that question for you, but it's probably could be on the order of 10%. Yeah. Yes?
How does the RNA primer overcome the obstacle of the 3' to 5' synthesis?
There's no 3' to 5' synthesis.
But it's moving that direction.
So RNA synthesis goes 5' to 3', just like DNA does. Yep.
Question. So, for example, in a circular DNA of, okay, so for example, lagging fragments, there is Okazaki fragments, and then once we have a RNA, DNA, then polymerase 1 comes, can it just chew RNA and this complete piece of DNA before the next RNA?
So the question is, again, related to the one earlier, which is how long will that DNA polymerase chew? And again, it falls off. So it's a random thing falling off. There's no set distance it's going to go. Okay? One more question, and I think we're not going to have a song today. That's fine. Go ahead. The dimers of the DNA do not separate from each other, no. They don't go different directions. They only go for a short distance, held together. We'll see that next time. Okay, I'll see you guys on Monday.